Welcome, everybody, to our first webinar with the University of Georgia. And today's topic is understanding the scientific method through the lens of plastic pollution. First of all, I want to thank the University of Georgia and our presenters today, and also want to thank the Herbert W. Hoover Foundation for their support with this whole project. Today, will be the first of the 3 webinars. So we're going to have one in mid-March. We're going to have another one in April. We'll get that to you a little later. Also, the intentions for this are for the students to get ideas for projects that they would want to carry forth into next year. If you're going to use them this year, that's great too. But also to get you a little jump start and maybe science fair for next year, because the Ohio Academy of Science, which sponsors a lot of the science fair ideas and things that go on with science fair in Ohio, they have quite a number of scholarships, and we think this will give you guys a really good jump start for getting ready for those projects. And without further ado, I'm going to turn it over to Catherine Youngblood from the University of Georgia, and we're going to get started.
Thank you, Dale, and thanks for the opportunity to be here to share a little bit about our work today. I'm going to get my slides up here. Alrighty. Can you guys see that all right?
Yep, looks good.
Perfect. All right. So I wanted to give a little bit of background by backing up and talking about our collaboration with Stark County back in 2022 in our Ohio River Earth to Oceans project, where we tracked litter along the Ohio River. So this kicked off in Earth Day of 2022 and wrapped up on World Oceans Day. The idea was to help symbolize that connection between what happens in communities upstream and how that might impact the ocean, especially in regards to plastic pollution. In total, we had researchers and volunteers log nearly 20,000 litter items in 4 different communities along the river and in the watershed. So we were able to characterize what litter looks like in those different places, including in Stark County. And I'll show you very quickly. Our next webinar is going to cover this in a bit more depth, but wanted to show you what we use the Debris Tracker Citizen Science app to record litter that we found in the communities along the river. The next slide. In Stark County, we did find the highest litter densities of all the cities that we worked in along the river. So that means that Stark County is really a place that's really ripe for continuing to collect this kind of data, as well as looking for upstream interventions to this problem. Most of what we found in Stark County was plastic, and that's why I'm excited for you guys to get to learn a little bit more about research on plastic pollution from our speakers today. So today we're going to get to hear from Taylor Maddalene, who's a PhD candidate in environmental engineering and the director of the Circularity Assessment Protocol at the Jambeck Research Group at the University of Georgia. Taylor got her bachelor's in environmental science from the University of Virginia and her master's in marine biodiversity and conservation from Scripps Institution of Oceanography at UC San Diego. So her background's in marine ecology, conservation, science communication, and pollution prevention. And then you'll also hear from Karina Carrasco, who is an undergraduate in environmental engineering, who's been working with plastic-eating mushrooms for almost a year now, and she'll be telling you about her project. So without further ado, I will go ahead and kick it over to Taylor.
Thanks, Catherine. That's a lovely introduction. I'm just going to share my screen as well so you guys can see my slides. Let me know if they're popping up okay. Excellent. Great. So I am going to be talking to you guys a little bit more about some of the research that I've done related to plastic pollution. We're going to be talking trash for the next couple minutes, so I apologize in advance for all of the trash-related puns that I'm sure I will inadvertently drop in here. But as Catherine said, I am a PhD candidate in environmental engineering at University of Georgia. I also serve as the director of what we call our Circularity Assessment Protocol, which I will explain a little bit more in here. But we do research all over the world related to plastic pollution, waste management, and pollution prevention. And it's taken us to all corners of the world and looking at all different kinds of contexts and cultures. So come on, there you go. It also needs coffee this morning. So just a little bit more about my background and how I got to where I am. So as Catherine said, I did my undergraduate in actually in environmental science. And so I had always been a lover of the ocean. I am happiest when I'm in, above, under, around the water. And so I decided that I wanted to dedicate my career to ensuring that, future generations had that same magical experience. So I got into marine ecology, marine biology, and then I sort of took a slight turn towards from studying pollution to preventing it from getting into the oceans in the first place. I did my master's degree at Scripps Institution of Oceanography in San Diego in marine biodiversity and conservation. And so I've been kind of all over the place. And after every degree that I got, I said I would never do any more. And here I am getting my PhD. So, the education is not linear. And really my biggest recommendation for students is to get as much experience in everything that you possibly can. It's just as valuable to know what you do want to do as what you don't want to do. And every experience is useful. So why did I get into plastic in this first place? How did I go from, my scuba diving to dumpster diving? So as you guys are probably aware, plastic pollution has become a really hot topic over the last couple of years. And part of the issue is that the largest proportion of the plastic that we're making are single-use products. So that hot pink bar at the bottom there on this graph that shows the growth of plastic over time, that's packaging. And that's usually intended to be used by the consumer for less than 10 minutes and then discarded. But plastic is designed to last forever. It was a miracle product that came about in the 1950s that can take on any characteristic, can take on any type of form, function, and then it essentially lasts forever. But we're increasingly making it for single-use products that mostly end up in landfill or the environment. So our research group did some research back in 2015 looking at, all right, so we know that we're making exponentially more. Where is it all going? And our research found that almost 80% of plastic that's ever been made ends up in landfills or in the environment. So that's something that is designed to be, again, like a miracle product to last forever that ends up being trapped in a landfill or polluting our environment. Less than 10% globally at the time had actually been recycled, and only about 12% had been incinerated. So if you're thinking about it, this is a challenge of scale that we're producing more and more and more. But the problem is that it's also ending up in all of the wrong places at the end of its life. Some of those places are out in the environment. So this is a mishmash of a couple different data sets that National Geographic came out with in 2018. But all of those pink little circles that you see out in the ocean are known concentrations of microplastics out in the water. So if you think about it, Catherine was talking about how we were looking at the Ohio River watershed. If you're within a watershed and a piece of litter drops on the ground there, that means that it is all going to flow roughly to the kind of the same location, which will ultimately flow out into the ocean. And all these yellow parts that you see jumping off from the continents are known concentrations of plastic that are coming out from river systems. Once it gets out into the ocean, it's really hard to collect, really expensive to clean up, and it travels around and gets caught up in ocean circulation, which is why you see those concentrations in the middle of the oceans. And so, like I said, we're producing more and more. It's increasingly used for single use, and it's ending up in all the wrong places. And so I was studying ocean pollution for a long time. And then when I got my master's, I ended up studying on this tiny island here in the absolute middle of nowhere. And what you think about when you think about a Pacific island in the middle of nowhere is you think about beautiful blue water, clear beaches, white sands, images like this. And to be fair, we definitely did see a couple of really spectacular images like this. But what we also saw was this all over the island. And this is a very remote location that doesn't have a lot of people there. And if you look at the trash on this island, on this beach, it comes from Japan. You can see brands from the United States. You can see brands from Australia. The way the ocean circulation works, once it gets out there, it ends up in these concentrated spots. And so I had this sort of moment where I'm here studying pollution in the ocean, and I see a scene like this, and it just feels so tangible and solvable. And so then I started working on, preventing waste from getting to this point in the first place. And you'll hear me in this presentation talk about circularity, or circular economy, which is a phrase that gets thrown about a lot right now, as a solution to plastic pollution and a solution to waste generation originally. So I was gonna ask folks if they had any ideas about definitions. Maybe this is a moment where teachers can ask students what this actually means. But I wanted to give you a glimpse into what we mean when we say circularity or circular economy. It's really related to the concept of circularity in ecology. So if you think about a natural system, every output becomes an input. The concept of waste in and of itself is basically obsolete. When we say circularity, we are not just talking about recycling. Recycling plays a role in this system, but it cannot solve the problem on its own. It really needs to be part of a larger chain of solutions. And when we think about circularity, especially from an engineering perspective, thinking about cities, or in my case, island nations, as complex systems that can optimize circularity is really useful when we think about research, because you can think about it like a contained system. So there are lots of different intervention points when we think about the plastic value chain and the life cycle. If we want to stop it from ending up on that coastal beach in the middle of the South Pacific, there are lots of different points along its life where we can stop that from happening. All the way from how it gets produced, how the product is designed, how waste is generated and used in the first place, how it's managed and collected, and then ultimately kind of capturing it once it's in the environment before it gets to the point of reaching out in the ocean. And ideally, all of these pieces kind of speak to each other. So what you're learning about what's ending up in the environment can help you think about, why did it end up here in the first place? And what do we need to redesign or rethink in the system to prevent that from happening? So you heard me mention the circularity assessment protocol earlier in the presentation. So this is essentially a methodology that we put together that takes that linear life cycle of plastic and turns it into a circular method that we can analyze with qualitative and quantitative data. So there are 7 different points along this spokes here, similar to 7 different stages of the life cycle. And if we look at a city or an island or a specific system, we can look at all these different pieces, and it helps us fill in the gaps of how we make it a more circular system. And we've done this so far in over 50 different cities in 13 different countries. The generation of waste is a very basic human function, but it looks very different in different places. And you can learn a lot about cultures and practices and people when you look at what ends up as waste and why. And there's a lot of amazing solutions out there. So it's been a really interesting process taking this around the world. So I'm going to end with a little bit of a deep dive, no pun intended, on a location that I do a lot of work in the Caribbean. This is an island called Dominica. So this is an island in the Lesser Antilles. This is a location where we've been implementing that circularity assessment protocol, but then also adding in some environmental factors, looking at different types of ocean pollution, marine pollution, and things like that. So this is a place where I've been doing a lot of my PhD research, and I'm just going to explain a little bit further how the circularity assessment protocol is getting applied to a place like Dominica. So if we think about input, the first stage in the cycle, what are we trying to get at here? What are we looking at? And what you can do is think, if you work your way through the life cycle and think about, what is the question that we want to answer here? What do we want to know from this stage of the life cycle? And for us, the question for input is, what products are sold in the community or the city or the island or location, and where are they coming from? And then we can think about what kind of methods are going to get us to that question, that answer. So what we do is we do surveys of convenience stores and grocery stores, and we look at all the different products that are there for really common things like drinks, chips, candy. And then we look at their manufacturing locations and their parent company locations. So for example, you might see that something comes from Coca-Cola. Their parent company is headquartered in the US, but they've got manufacturers that are much closer down in the Caribbean. So you think about, where is this product physically coming from? How did it get here in the first place? Who owns this market for these products in this place? And that helps us understand where we could potentially work with producers and manufacturers to talk about, taking their products back, rethinking the way they're delivered. There's lots of kind of economic opportunities in that. The next one we look at is product design. And the question that we're trying to get here is, what materials, formats, innovations are found in products? So what is stuff made of physically? And if we're trying to figure out what that is, again, we do lots of store surveys for convenience products, like I just said. We'll also look at grocery stores. So we'll go in and look at things like milk, rice, greens, eggs. Are they mostly packaged in styrofoam? Are they mostly packaged in film or cardboard? And then we also look at restaurants. So we'll go to food vendors and restaurants and say, what is the to-go cup made of? What are the to-go food containers made out of? Because these are the things that most commonly end up in our municipal waste systems, and are potentially likely to end up in the environment. And then we also look at bag surveys. So from all of those convenience, grocery, restaurant stores, we'll look at what type of grocery bag or single-use plastic bag that they give out, or if they've got reusable options. So moving swiftly on to use and reuse, if we're looking at this stage of the life cycle, what we want to know is, what are community trends around the use and reuse of product types? So are there any reuse and refill stores? Some of you might have things like bulk stores where you are, or places where you can bring your own container or bring your own bag. Are there any plastic alternatives? So this is actually a wall of compostable plastic that's available in a grocery store down there. What does the cost comparison look like between those options and the availability of those different alternatives and reusable things and refillable things? And then are there any policies related to that? So we also do a series of stakeholder interviews. So we talk to people about their perceptions around these issues too. Do you feel like you have the opportunity to bring your own bag? Do you feel like there are, you're getting an access to waste management and collection that you need? So this is kind of quantitative and qualitative. And then for collection and management, we're looking at how much and what types of waste are generated, how is it collected, and then what happens after it's collected. So for this, we'll look at things like waste characterization studies. And this happens sometimes at landfills or dump sites or recycling centers, where they see, what are we bringing in? Like, what proportion is plastic versus metal versus glass? What's coming from households versus businesses? And then we look to see if that actually matches the waste management infrastructure that's available there. So for example, one problem that we see in a lot of cities is that you'll get a lot of new, innovative, compostable plastic materials when we look at the product design section, but there's not industrial composting facilities to deal with those. So they're still ending up in landfill. So a lot of this is kind of problem solving and matchmaking to see if what we're finding upstream in input and product design and use is also matching the capacity and ability to manage it downstream. And then last but not least, we look at leakage. So like I said, we're looking at what ends up in the environment, how did it get there in the first place, how do we prevent it from getting there? And we do this in a couple different ways. One of the ways that we do this is similar to what Catherine showed from Stark County. So we use the debris tracker app, and we go through and do randomized litter surveys across the locations. So this is an example of some of the litter surveys that we've done in Dominica. And it's basically the same way that we do it in lots of other places. And this is all freely available and online. You can go onto the debris tracker website and zoom in on Dominica the same way that you can zoom in on Stark County and see what's out there. And then for Dominica, because we were also concerned about the marine environment, we did some offshore litter surveys. So this basically involved me being in a small boat and going very slowly just offshore and seeing what type of floating litter we could see on the top of the water, and then logging that in a very similar way on debris tracker. And this is the kind of results that you get. The other way that we look at this specifically for Dominica is that we were also wondering about what happens once it enters the environment. What happens once it enters the ocean? What does that actually look like? And so we did a couple different methods there to answer that question. We set up a river litter trap. So this is this big orange kind of floating thing that you see. Basically, the intention there is to document what waste is coming through the river system before it enters out into the ocean to see if that's a major source or not. We also did water microplastic samples. So we took samples of ocean water offshore, and then we were able to use some analysis to see if there were any microplastics in the water, and then what type of microplastics. So for example, if you're seeing a lot of PET, does that match with the litter surveys that we're seeing on land where there's lots of bottles? And then last but not least, we also did microplastic analysis of different types of fish guts, because that's another major concern, especially for island nations, is that they depend really heavily on the fish for food. And so they want to know what's actually in the fish, what type of other environmental concerns they have. So we really tried to cover all of our bases of, how is it ending up in the environment? Where is it coming from? How do we stop it from getting there in the first place? And what are some of those downstream effects that we need to be aware of that can help drive that upstream prevention? And one of the things that I really enjoy about working in plastics right now, but then also working with small island developing states right now, is that there's a big UN treaty in negotiation right now related to plastic pollution. This is the first time that we've ever had something at this level to internationally and in a collaborative and concerted effort address plastic pollution. And everyone from the US down to, small countries and everything in between has got a voice and a stake in this argument. And it's really been interesting being the vessel that can help provide some of that data to support what countries want to see. So one really exciting thing coming out of this work in Dominica is that we're able to use that circularity assessment protocol data to inform things like a national action plan, or to advocate for different policies that you want to see that's going to help with things like import of products or management of products. So working in this plastic space is really exciting because all of our work is very relevant, but it also has a tremendous opportunity to actually inform positive change going forward. So it's an exciting time to be in this space. There's lots of interesting things happening. And if you're interested in science, policy, business, the environment, there's lots of different ways to get into this space and to make a difference. So it's an exciting time. And that is all that I have on some of our research and our work. I'm happy to answer any questions. Please check out our website that's got more information on our methods and some of the reports. And I will pass it back to Catherine.
Thank you, Taylor. That was great. That was really fun to get to hear more about your work. Karina, we would love to hear from you next.
Hello. Can you guys hear me?
Here you go.
Okay.
Yep.
Okay. Sorry, I've never used this.
No worries.
Oh no.
So the present now button's in the middle of the bottom screen, and then there should be an option to, if you look on the right-hand side of that, once it pops up, there should be an option to share your entire screen, and that should show your slides. That might be that was the way I was doing it anyway.
Okay.
Looks good, Karina.
Yep.
I'm not sure if you guys can see me because I can't see you guys or what I'm sharing, but
we can see you and your slides, so we're good.
All right. Okay, so oh no. So thank you for having me today. I'm excited to share with you guys what I have for you guys today. So I'm going to talk to you guys about the research that I've conducted on household plastic with the help of fungi. So when a tree in the forest or the leaves hit the ground, we know this signifies the end of a cycle, but what's really happening is the beginning of a new cycle of rebirth. Without the work of fungi, there would be no decomposition on Earth, leading to towers of organic matter. If a falling tree doesn't rot to make soil, new trees wouldn't be able to sprout. Decomposition is just one of the many things nature's recyclers are capable of doing. So just a quick introduction to kind of like set the scene for what I'll be talking about. And scientists have found multiple species of fungi that are able to degrade plastic and feed on it by turning it into organic matter. This is referred to microremediation. And so companies and inventors around the globe are tapping into the fungi kingdom to try to understand mushrooms a little bit better. And they're being used to replace things like leather, shoes, handbags, packaging materials, furniture, and even car interiors. And there's actually a Dutch company that created the first living coffin, as they call it, which is literally a coffin that you can buy that's made out of mycelium in as little as 7 days, and then it takes 45 days after you bury it to biodegrade, which I think is pretty cool. But one of the bigger companies that have been implementing mycelium technology is Ecovative, and one of their more popular products are their leather that they have created. And this is kind of like an example of what they offer. And so it's essentially all made out of mycelium, which is just the root-like structure, or like the web of a fungi that essentially allows everything to be consumed as food. And yeah, they've also have found ways to implement different textures and finishes on the leather to give it more of like a look of leather, which obviously later down the road, when it comes to the end of its life, it's able to biodegrade and you don't have to worry about it anymore. So mushrooms are capable of many things, like I said, but there's still more than 90% of them that still remain unknown to science. But the biggest thing here is that they can eat plastic. So last year I took an environmental engineering design methodology course, which was ran actually by Dr. Jambet and Taylor. So I ended up designing a solution for at-home plastic waste. And then I'll just talk about the first experiment that I conducted. It was, and how I went about planning it. I decided to initially start with P. microspora, which is one of the fungi, and then also the other one was oyster mushrooms. But I decided not to talk about the microspora because it was just too much. Like, I didn't want to bore you guys, but yeah. So I ended up using just the oyster mushroom later down the road. And apart from them being carnivorous, oyster mushrooms are saprophytic fungi, which means that they release specific enzymes to break down materials such as tough plant fibers and turn it into energy. This aspect of fungal metabolism is key in the field of microremediation. And that's because plant fibers such as lignin have similar molecular structure as certain environmental pollutants. And there have been multiple experiments conducted using fungi, but the biggest one is to break down plastic. So this was what I had come up with. Sorry, my throat is really dry. Okay, sorry. I was sick last week, so I'm still trying to recover, but yes, so this was the design that I came up with, which is a possible way to apply at an at-home scale. And the experiment that I conducted was made in a way to support my prototype, to back up the design. And this is kind of just an overview of the experiment that I did the first round, which was just inoculating brown rice bags, because that was just the most financially, the best financial way I could conduct the experiment. I got this syringe from the website, inoculated the bags, which then once they were fully colonized, like on the first picture, I was able to open them up and then incorporate the plastics inside, which I ended up doing polyurethane. And I did, like, on the side of, except for the apart from the bags, I did on the side on some tubs, I did some PET plastic, which, because of, I was just curious to see what would happen, because I didn't really find much information on people like using PET plastic in oyster mushrooms. So after that, I ended up adding a layer of cocoa coir, and then you can just see the mycelium kind of just take over, and it's just being really happy. And then it took a while for them to fruit, which is the last picture there, but I did learn a lot along the way that later helped me when I conducted the next experiment. And so after the bags fruited, I was able to see what exactly they did. And this kind of shows the packaging foam, how it made little like indents, and it like significantly definitely changed the shape, and it just looked completely different from when I first put it in there. And well, for the other plastics, which was a PVC pipe, wasn't really that much of a difference. And same thing with the PET plastic, it just looked the same as when I put it in. So I decided to, for the second experiment, I asked myself what I could have done differently, and decided to take a different approach to focus more on directed evolution. So if you think of a fungus living in the woods and growing on dead wood, then out of nowhere, they just take their food away. They can't just get up and leave like the rest of the animals to go find new food. So they're forced to adapt. And fungi are actually able to shift their metabolism to produce completely different enzymes to alter their structure and appearance. So taking away their food source and causing them to stress will lead the fungi to morph into a strain that can digest other things like tissue of living things. And it sounds scary, yes, but humans, our bodies are too hot for fungi to survive. So, which is why historically they have only been known to grow on nails and skin. So, well, for now, so hopefully it stays like that. And this is the step that I took figuring out a new plan. I wanted to start with placing liquid cultures on nutritious petri plates, and slowly start taking away their food and introduce plastic. And before I got to that point, in phase 1, I started with many strains and narrowed it down to one plate, which is the one that I'm currently working with. These are pictures that I took along the way, where I started with 10 different plates, and I'm showing you guys just the progress of one of those 10 plates. And this also like ensured me that, oh my goodness, sorry. This also ensured that the liquid culture that I received was not contaminated, which is a very big thing when you're dealing with anything in mycology. That's the biggest thing, just to follow aseptic procedures, because once you contaminate anything, it's just, the whole thing is just done for. You can't use it. So one of the dishes were once the dishes were fully colonized, I cut out little circles of each plate and then transferred them to 10 neat plates with different agar. And this was when I was able to see which of the 10 plates had the strongest genes. So I was, so on the bottom left, you can see I just showed one of the 10 plates. So by the end of this phase, I had 10 plates that looked like the plate on the bottom left. And I tracked their diameter growth and determined which one was the most aggressive and vigorous of the 10 plates. So for 10 days, I measured and recorded the diameter of them. And as you can see here on the data, each color or line represents a different date. And if you line it up on the bottom with the plate, you can see the rate that which they're growing. And for my results, I ended with plate number 9 having the highest growth rate. This is the one that I introduced the plastics to. And there was a lot of things that I was really shocked by along the way. In the second experiment, I encountered some unexpected results. The expectations that I had were very low for them to fruit, but considering the first experiment, but they didn't fail to amaze me. While growing them, the petri dishes actually ended up fruiting, and it's not really expected for them to pin or fruit. Yet they did give me little baby mushrooms. The thing is, what's I guess I couldn't really wrap my head around is that normally fungi need some type of nutritious substrate to prepare them for fruiting conditions, which includes fresh air exchange, lighting, specific temperatures, and humidity. So I didn't expose the dishes to any of this except for the lighting that's coming in from the lab. But before I knew this was going to happen, I had already started and prepared fruiting bags to see if the dish number 9 that I ended up with had the fruiting DNA or not. Though it wasn't really necessary because the dishes answered the question for me, it was still fun to see them grow and give me a pretty good flush. And then this is this is the results that I got, which was really rewarding in the end. And today I'm still in the progress of introducing microplastics to the mushrooms to essentially slowly train them / encourage them to make plastic their new favorite meal. So more research is needed for sure to determine the safety of these plastic eating mushrooms, whether for ingesting or waste management. But until more research is conducted, our current solution to eliminating our plastic waste is reducing our consumption and production of all plastics. And yes, that will be it for me. Thank you.
Thank you, Karina. So much fun to get to hear more about your work. That was awesome. Thank you so much.
Thank you.
Can't take the screen sharing from you. All right. All right. Oops. Not where I wanted to start from. There we go. Okay. Well, thank you so much, Karina, and thank you, Taylor, for sharing more about your research. And hopefully all you guys listening found that interesting and inspiring for thinking more about the science that is needed to understand more about plastic pollution, as well as other environmental issues. So we did put together a few extension activities, some additional reading, some questions to get you thinking, some more lessons to explore. We'll drop this link under this video as well, so you'll be able to access it. And I also wanted to mention that our next webinar will happen at the same time, 9 a.m. on March 13th. And we'll be talking about interdisciplinary data collection and analysis. So looking at citizen science, geospatial data on litter, as well as insights on how to conduct interviews and collect qualitative data from our environmental anthropologists on our team. So we hope you can join us for that. And thank you everyone for your time and attention today.
Thank you, guys. I really appreciate it. I want to thank Catherine, Taylor, and Karina for the presentations today. And also, once again, thanks to the Herbert W. Hoover Foundation. We are recording this one, so this will be available through our website, and we will get that word out to all our teachers and our students.